(2b) Organic chemistry. Chemistry can be divided into two very broad categories: organic and inorganic chemistry. These terms are actually holdovers from earlier times, when it was thought that organic substances came from only animals and plants, while inorganic substances came from mineral sources, a belief which is not totally accurate. Organic compounds are those that contain covalently bonded carbon atoms, with a few exceptions. Their unique properties are based on the carbon atom and its unusual bonding characteristics. Every organic compound has carbon in it, usually in conjunction with large quantities of hydrogen and frequently with oxygen. Living organisms make organic compounds that are required for life. Many of these compounds are very complex and large molecules, though some of the simpler ones have been synthesized in laboratories. Under normal circumstances, most organic compounds are the result of biosynthesis, the putting together of substances by living things. Biosynthesis is the process by which assimilation is accomplished and is essential to life. Occasionally, headlines say that life has been created in a laboratory. These misleading headlines are written to get attention. A close reading of such an article, if it is written accurately, reveals that the new development falls far short of creating life in a test tube. Usually, scientists have simply manufactured another biochemical compound by use of an elaborate apparatus. To accomplish even this, they must use large amounts of energy, special procedures, and elaborate equipment. Each time scientists employ sophisticated techniques to synthesize these compounds, they actually indicate how unlikely it is that life could spontaneously generate in a test tube, much less in a prehistoric pond. To create life, not only must one organic compound be made, but literally thousands of different ones must be made in large quantities, all at the same time, for many of them are very unstable on their own, and all in the same place. Next, these materials have to be given life. Will life just happen if all the chemicals are right? Even those scientists who attempt to create life in a test tube admit that their chemicals need some special starting force. Some evolutionists have suggested that a bolt of lightning in a prehistoric pond started life. Regardless of thunderbolts or special jump starts, life is more than the proper alignment of the correct chemicals. Even unbelieving scientists must admit that there is a great contrast between a living organism and that same organism after its life has ended. With our fallen minds, we cannot fully comprehend the secret to life. But a Christian worldview requires us to acknowledge that the Creator plays a key role in both the original creation as well as the continuance of life. Whether God chooses to use only physical processes to accomplish this, or instead use supernatural forces beyond our comprehension, it is still in His hands. Is it possible for a living thing to be assembled from non-living substances in a laboratory? At the time of this writing, it has not been done. Life, the Scriptures tell us, is the creation of God. Isaiah 42:5. But the Bible does not tell us that humans will not be able to put together a living thing? If, by using elaborate equipment, skill, and other man-made chemicals, scientists do form a structure that can be called alive, have they disproved spontaneous generation and evolution? Number. They have simply shown that man-made chemicals, man-made equipment, and intelligence is necessary. They have not disproved the concept of a special creation of life or discovered exactly how God created life. Also, any life that humans could make in a lab would be made differently than the way God made life. God makes life out of nothing, while humans have to use materials already created by God. 2.4 Objectives. Describe how carbon can form so many different compounds. List the three main functions of organic compounds in living organisms. Key term: organic compound. 2.4 Organic compounds. A carbon atom has 4 electrons in its outer shell that will readily form covalent bonds. Carbon is unusual because it will readily bond with other carbon atoms, forming long straight chains, branched chains, or even rings of carbon atoms. The chain or ring upon which the remainder of the molecule is built is called the carbon backbone. As Figure 2-17 shows, a great variety of organic molecules can be formed. Carbon will also share more than one electron with other atoms, either another carbon or a different atom. If a carbon shares two electrons, it is called a double bond. A triple bond occurs when three electrons are shared with another atom. These different types of bonds will affect the shape of the organic molecule, and shape is very important in organic chemistry. Glucose and galactose, introduced in subsection 2.1, have identical molecular formulas, but it is their structural formulas, their shapes, that determine their unique characteristics. Organic compounds perform various functions for living organisms and can be categorized as follows. Structural, compounds that are used as building blocks of a cellular or extracellular structure. Enzymatic, compounds that are enzymes or that help enzymes in their functions. Storage, compounds that store energy, other substances, or information for future use. Many of the organic compounds perform combinations of these functions. The large number and great variety of organic compounds make grouping them difficult. They are often classified into four groups: carbohydrates, lipids, proteins, and nucleic acids. And 2.5 objectives. Describe the basic structures of carbohydrates and lipids. Discuss the various functions carbohydrates and lipids have in living organisms. Differentiate between saturated and unsaturated fats. Compare the properties, hydrophilic and hydrophobic. Explain the relationship between monomers and a polymer. Key terms: carbohydrate, monosaccharide, glucose, disaccharide, polysaccharide, starch, glycogen, cellulose, lipid, fatty acid, hydrophilic, hydrophobic, triglyceride, saturated, unsaturated, steroid, monomer, polymer. 2.5 Carbohydrates and lipids. No matter where they are found, in plants, pigs, or people, carbohydrates and lipids are the same, although their amounts may vary. Both are organic compounds, both store energy, and both are important molecules in many structural components of organisms. Molecules of carbohydrates and lipids can be either quite small or very large, consisting of thousands of atoms. Yet, they are also different. Many of the less complex carbohydrates dissolve in water. Most lipids do not. Gram for gram, lipids store more energy than carbohydrates. Carbohydrates. Carbohydrates, K-A-R-B-O-H-Y-E-drates, are organic compounds that contain carbon, hydrogen, and oxygen. Generally, the carbohydrates found in living things have twice as many hydrogen atoms as they do oxygen atoms. Monosaccharides. The basic units of carbohydrates are monosaccharides, MAHNUHCK-rides, or simple sugars. A monosaccharide may contain as few as 3 carbon atoms. The monosaccharides that contain 5 or 6 carbon atoms are important building blocks of other organic compounds. A common 5-carbon sugar is ribose, a component of nucleic acids. Glucose is a 6-carbon sugar manufactured by plants. It is also the sugar that the blood transports to cells in humans and in many animals. 2 other simple 6-carbon monosaccharides are important in biology: galactose and fructose. Disaccharides. Monosaccharides can be joined together in chemical reactions to form a disaccharide, disaccharide. In Figure 2-18, a glucose and a fructose molecule combine to form the disaccharide sucrose, a common sugar found in many foods and often called table sugar. Because a water molecule is released in the synthesis reaction that forms a disaccharide, it is sometimes called a dehydration synthesis reaction. When a cell needs the monosaccharides from which a disaccharide is made, enzymes will combine the disaccharide and a water molecule to separate the disaccharide into monosaccharides. A reaction such as this, in which water is added to split a molecule, is called hydrolysis. Polysaccharides. A polysaccharide, pal-E-saccharide, is a large molecule of monosaccharide units. Using specific enzymes, living things can build up or break down polysaccharides. 3 polysaccharides are particularly significant. Starch is one of the primary substances that plants store as food. Starch from corn, potatoes, wheat, rice, and other grains constitute the major energy source for humans. Glycogen is often called animal starch. The starches a person eats are broken down to simple sugars and then taken, along with the other sugars in his diet, to the liver where they are made into glycogen for temporary storage. Glycogen is a branching chain of glucose molecules. Cellulose, SLU-lose. Molecules are much larger than starch molecules. They are made of long chains of glucose molecules bonded in an alternating arrangement. The long cellulose molecules account for the strength of plant cell walls. Plant fibers that form cotton, wood, paper, and many other products contain large quantities of cellulose. Lipids. Lipids are a group of organic substances that are only slightly soluble in water, but are very soluble in organic solvents such as alcohol, ether, acetone, and chloroform. Lipids are most often structural, but they also store energy. Because lipids occupy less space than starches, humans and animals store their excess energy as lipids. Plants, however, add to their size by storing starches. The most abundant lipids are fatty acids, which are unbranched chains of 14 to 28 carbon atoms that have a carboxyl group, -COOH, added to one end. The carboxyl end of the molecule is polar and is attracted to water. It is hydrophilic, HYEE-DRUFIL-IK. The other end is repelled by water and is termed hydrophobic, HYE-DRUFOH-BIC. This unusual characteristic permits fatty acids to be used in specific ways in cells. The importance of these characteristics will become clear as this study progresses. Fatty acid molecules can be broken down by enzymes into many 2-carbon molecule pieces. Each of these molecules may eventually release usable cellular energy, making fatty acids a good energy source. The body's most abundant type of lipid is triglycerides, or fats. Triglycerides are formed by combining 3 fatty acid molecules to a molecule of glycerol, glycerin, a 3-carbon alcohol. If each of the carbon atoms in a fatty acid molecule, except the end ones, has 2 hydrogen atoms attached to it, the fatty acid is said to be saturated. However, should one or more of the carbon atoms be double bonded, there are fewer hydrogen atoms attached to the carbon backbone. This type of fatty acid and molecular structure is called unsaturated. The straight chains of a saturated fatty acid allow them to stack tightly together. Therefore, at room temperature, a saturated fatty acid will be a solid. Examples include butter and lard. The double bonds in the unsaturated fats cause bends in the chain and prevent a tightly stacked arrangement. These molecules will exist as liquids at room temperature. Lipids that are liquid at room temperature are called oils. Corn oil, olive oil, peanut oil, and coconut oil are examples of unsaturated oils, as are most oils found in fruits, vegetables, and fish. Phospholipids are composed of 2 fatty acid molecules attached to a glycerol molecule. The 3rd carbon atom of the glycerol molecule has a phosphate-containing group attached to it. Phospholipids have a hydrophilic phosphate end toward water and a hydrophobic end that avoids water. This characteristic is essential in the formation and structure of the cell membrane, which will be studied fully in Chapter 3. Steroids have a carbon backbone of 4 different carbon rings, plus a side chain of different lengths. They are classified as lipids because they are hydrophobic, but they differ from lipids in their physical and chemical properties. Many steroids circulate throughout the body as chemical messengers. Sex hormones, estrogen and testosterone, are 2 such steroids. Cholesterol, the most common steroid, is found in the membranes of both animal cells and human cells. Macromolecules. Many of the molecules found in the cells of organisms are very large and are called macromolecules. Macromolecules are constructed from hundreds or even thousands of smaller molecules. The smaller units that make up a macromolecule are called monomers. When the monomers are assembled into the much larger molecule, it is a polymer. In some, but not all polymers, the monomers are identical. 2.6 Objectives. Describe the formation and structure of proteins using the terms amino acids, peptide bonds, and polypeptide chains. Recognize DNA as the primary nucleic acid and hereditary molecule in most organisms. List the 3 parts of a nucleotide and describe their arrangement. Compare and contrast DNA replication and RNA transcription. Key terms: protein, amino acid, polypeptide, nucleic acid, DNA, deoxyribonucleic acid, double helix, nucleotide, replication, RNA, ribonucleic acid. Transcription, RNA polymerase, 2.6 proteins and nucleic acids. Whereas carbohydrates and lipids are the same in different organisms, proteins, although sometimes similar in structure and function, are different. For example, human insulin, a protein that helps in food metabolism, is virtually identical to the insulin found in animals. Human hair protein, on the other hand, is different from the protein found in animal hair. Even among people, although one person's hair proteins are similar to another's, they are not identical. Proteins, it may be said, are what make a person unique. The uniqueness of an organism's proteins is the result of nucleic acids and enzymes. Nucleic acids contain the code to manufacture the proteins. Enzymes, which are themselves proteins, help the nucleic acids function properly. Thus, proteins and nucleic acids are codependent. Proteins. Proteins consist primarily of carbon, hydrogen, oxygen, and nitrogen, but they sometimes include phosphorus, sulfur, and a few other elements. They come in a wide variety and are used either as enzymes or as structural building blocks. Some are very small, but some protein molecules are large enough to be seen under high-powered microscopes. The basic building amino of a protein are amino acids. There are about 20 different amino acids. An amino acid contains an amino group -NH2 and a carboxyl group -COOH, both attached to a carbon backbone. Different side groups, some simple, others complex, attach to the carbon of the amino acid. Some proteins are composed of only 100 connected amino acids, but several thousand can line up to make some of the larger proteins. When two amino acids line up and the proper enzymes are present, a dehydration synthesis reaction occurs, removing a water molecule and forming a peptide, PEP-tide bond between the amino acids. A peptide bond links the carboxyl group of one amino acid to an amino group of another amino acid. A protein, then, begins as a polypeptide, peptide chain of amino acids. When the proper sequence of amino acids is arranged, the chain twists and then folds back upon itself. Certain amino acids bond together in a specific pattern that is determined by the sequence of the acids. The polypeptide chain of amino acids now has a three-dimensional arrangement. It is this shape that forms the active sites of enzymatic proteins. As many as 4 different levels of organization can be seen in the structure of proteins. The lowest level is the basic order of amino acids in the polypeptide chain. The polypeptide chain may then be coiled or even folded in a sheet-like arrangement. The coiled and/or folded structure itself is folded and held in a precise shape. Several of these reconfigured polypeptide chains, called subunits, may lock together to form a large protein. If some of the amino acids are out of sequence, the shape of the protein molecule may be altered. Should this change of shape affect the active site of an enzyme, the enzyme may not react properly with its substrate. Heat, radiation, ion concentrations, and other environmental factors can also affect the bonding of amino acids to other parts of the amino acid chain. When the bonds break between the amino acids that give the protein its three-dimensional shape, the shape of the protein molecule may change. If this change affects the active site of an enzyme, it may no longer function as an enzyme. These changes in shape may also affect the ability of a cell to use the protein as a building block for other structures. Proteins often work together with other proteins or other substances. Enzymatic proteins may require coenzymes in order to function. Some proteins combine with lipids, lipoproteins, carbohydrates, glycoproteins, or even other proteins to form a structural component. Many hormones are lipids joined with proteins. Nucleic acids. Although scientists have long known that proteins are different in various organisms, they did not discover until just before World War I that nucleic acids, not proteins, are the hereditary material. In other words, in order to transmit its characteristics to another cell, a cell must pass nucleic acids to that cell. Nucleic acids, in the form of DNA and RNA, found in the cell nucleus, direct the activities of a cell by guiding the formation of both structural and enzymatic proteins, because they contain the information necessary for the manufacture of the organism's proteins. Nucleic acids determine its characteristics. The way the nucleic acids direct protein manufacture and the exact method of transmitting this information to the next generation of cells are important concepts discussed in later chapters. This section examines what nucleic acids are. The structure of DNA. DNA deoxyribonucleic, DEE-AHKC, ribo-no-nucleic acid is the primary nucleic acid in most organisms. It contains the information that is needed for almost all of a cell's activities. By the time of World War II, scientists had analyzed the components of DNA, but were confused about their exact arrangement. To understand how DNA functions, it was necessary to form a working model of its components. In 1953, James D. Watson, an American biologist, and F.H.C. Crick, a British biophysicist, assembled information about DNA's components. Using information obtained from X-rays of DNA, they created a DNA model. Watson and Crick received a Nobel Prize in 1962 for their accomplishment. Although it has been slightly modified since then, the basic model is still consistent with all the information known about DNA. Watson and Crick's model is a double helix or spiral, consisting of two strands attached at regular intervals. Visualizing a flexible ladder that is twisted on its long axis gives a good idea of what a DNA molecule looks like. The basic understanding of the DNA structure must begin by mentally untwisting the DNA molecule and examining its component parts. DNA is a double-chain polymer of nucleotides. A nucleotide, nucleotide, has three components. Sugar. The sugar in DNA is the 5-carbon sugar deoxyribose. Phosphate. The phosphate groups in a nucleotide is a small molecule made of phosphorus and oxygen atoms. Base. Each nucleotide will have one of these 4 bases: adenine, thymine, guanine, or cytosine. Just as in the formation of polysaccharides and polypeptide chains, the nucleotides are put together by a dehydration synthesis reaction. The nucleotides are arranged so that the sugars and phosphates form one long side piece of the ladder, with the bases pointing off to one side, forming one half of each rung, step, of the ladder. The other half of the ladder is likewise formed by nucleotides. Figure 2-27 illustrates DNA base pairing. Look carefully at the bases. Each adenine is bonded with a thymine by 2 hydrogen bonds, and each cytosine is bonded with a guanine by 3 hydrogen bonds. When there is a thymine on one side, there's an adenine on the other, and when there is a guanine on one side, there is a cytosine on the other. DNA replication. The sequence of the bases in the DNA molecule can be compared to the sequence of letters in this sentence. In one order, the letters make words and have meaning. In another order, they may make different words and mean something else. In a different arrangement, the letters may make words in another language, or they may be completely senseless. The sequence of bases in DNA forms a specific code that directs the sequence of amino acids in the protein molecules that the cell produces. The message encoded within the sequence of bases in DNA must be copied so that it can be passed to the next generation of cells. This copy must be an exact duplicate of itself, even down to the sequence of bases. If the base sequence is not correct, an improper protein may be manufactured, or possibly no protein at all. DNA uses itself as a direct blueprint in duplicating itself. Visualize this process as the opening of a zipper. An enzyme moves down the DNA molecule, breaking the hydrogen bonds between the bases that hold the two sides of the DNA molecule together. The two sides do not close like a zipper, however. Instead, a different enzyme attaches free nucleotides to rebuild the missing half of the molecule. The final result is 2 identical molecules of DNA. Once complete, the sequence of bases is exactly the same in both strands of DNA. Producing 2 new DNA molecules from an old one is called replication. The enzyme systems necessary for replication are vast and intricate. RNA, another nucleic acid. RNA, ribonucleic, ribonucleic acid, is the basic nucleic acid for a few viruses. However, its function is to serve as the code for the production of proteins. There are three important structural differences between DNA and RNA. RNA is a single strand of nucleotides rather than a double strand, as in DNA. In RNA, the sugar is ribose, which has one more oxygen atom than the deoxyribose in DNA. Also in RNA, the base uracil replaces the thymine found in DNA. RNA is formed by DNA in a process called transcription. An enzyme called RNA polymerase, polymerase, starts at a specific section of the DNA molecule called the promoter site. From this point, the RNA polymerase moves down the DNA molecule, causing a section of it to separate. Other enzymes pair the proper RNA nucleotides to the corresponding DNA, forming an RNA chain. As the enzyme continues to move down the DNA molecule, the RNA chain separates from the DNA, and the separated DNA strands reform. This process is discussed in more detail in Chapter 4.